Thank you for joining Plexon for part 4 of our 5-part webinar series featuring the NeuroLight optoelectrode. This webinar and the question and answer session will be available on the Plexon website following the live broadcast. Today's speaker, Dan English from Virginia Tech, will discuss how his lab is using the NeuroLight optoelectrode to monitor and manipulate pre- and postsynaptic neurons. Thank you for joining us for his presentation titled Microcircuit Interrogation with Neuron-scale Optogenetics. If you have a question for Dan, please enter those questions in the Q&A section at any time. Please use the question and answer feature on Zoom rather than the chat feature for your questions. The speaker will address questions live during the question and answer session following the presentation.
Hello, everybody. Thank you for coming to the talk today. So we've been using these microelectrodes for quite some time. I actually started using these when I was a postdoc in Yuri Bujaki's lab. And so we were using them when Yusik Yoon's group was first developing them, or not very first developing them. That was with Ron Stark and Fan Wu, who we probably heard of in these talks. But when I started working with them, and Sam McKenzie, who was also did all this work with me that I'm about to talk about, who used to be also a postdoc in Yuri's lab, this was when they had started ramping up the production of these probes, and we were, they were really becoming the very low noise, super useful probes that they are today. And so today I'm going to talk about mostly work that I did with Sam in Yuri Bujaki's lab, and this is stuff that we're continuing to work on in my lab today. So the part of the brain that I study in my lab studies is the Ca1 region of the hippocampus, which is famous for many things, place cells and memory. But what we're really focusing on is the picture that you see on the screen in front of you right now. And so what we're looking at is what we really want to see is how small groups of neurons and even pairs of neurons communicate with each other in behaving animals to support all of the well-known spatial navigation and mnemonic functions of the hippocampus. And so I'm just going to briefly introduce to you the sort of behavioral tasks that we're thinking about and the kind of functions that the hippocampus is performing. And then, the talk is, our data, is going to be about how we use these micro LED silicon probes to interrogate the mechanisms supporting these functions. So I'm sure some of you know what place cells are, but briefly, as you watch this movie, there's a rat running around a little track. Each dot is an action potential on the screen related to the rat. That's where that action potential occurred, and each color is a cell. And so what you can see happens as the rat runs around this track is that there's a trajectory through space, and then there's a trajectory through a sequence of neural activity. And if you look at this on a different scale, so in the bottom left here, this is a single square environment and they recorded this many neurons. So each box is a neuron, and the color in each box is the firing rate of that neuron in that particular place in space. So you can see like the third from the left and the second down is, so this one here, that one is an interneuron which fires everywhere. But there's other neurons like this one and like the fourth down on the left and other ones that occupy a small part of space, but as a population, they cover the entire space. So the ensembles of place cells can represent different, as a population, the entire environment in which you're in. So it's like a cognitive map. But importantly, this isn't a simple response of the hippocampus to sensory inputs that, let's say, are varying through space. So the hippocampus is actually able to generate activity in these sequential trajectories on its own. So in this experiment that on the bottom right, an A is just sort of an example of a cartoon of, different place cells as the rat runs around, 1, 2, 3, 4, 5. They could all be getting inputs from another part of the brain that tells them when to fire in what place. Alternatively, they're obviously getting some kind of information from the outside world, but the actual sequence of cells, so which cell fires where, the alternative is that that's controlled by the hippocampus itself. And so this latter case turns out to be true. And this task from Eva Paselkova and Yuri Buzhaki, on the bottom right, you see the task is the animal has to run in the running wheel for a fixed amount of time, leave the box, run down this platform, and make a right turn. And then the next trial, make a left turn. I'm sorry, I don't know why it keeps skipping ahead. But it has to make a left turn. And essentially, it has to run in the wheel, run one direction, run in the wheel, run the other direction. So it has to spontaneously alternate and remember where it's going to go after leaving the wheel running each time. So it's not surprising that, again, each color here is a cell, that cells have place fields all over the track that tile these trajectories. But what is particularly interesting is what's going on in the wheel when the animal's actually running in the wheel, which is that that time is actually tiled just like space is tiled. So in B here, this is now each row is a cell, firing rate is color, high is red, low is blue. And so you can see that the time is also tiled by the hippocampus, not just the place. And so this is a clue that the local circuitry in Ca1 has to do something special in terms of generating these sequences. And so obviously hippocampus is very implicated in memory. So how these places are remembered. So to get an idea of that, we have to wait until the rat stops running. And this is the same video, but I want you to watch what happens when it reaches right here on the, at the very end here, like 1, 2, 3, this burst of cells is actually the same group of cells that were active along the behavioral trajectory, but now they're happening much closer in time. And it turns out that these trajectories are actually replayed during specific events. So in the bottom, in the middle here, what we see is essentially the same in the video. Here is a cell, the tick marks are obviously their spikes, and you see a trajectory of cells covering the space as you would expect if this is a cognitive map. However, before in red and after in blue on the right, there's these events in the field potential called the sharp wave ripple oscillation, which is about 150 hertz. And during these events, the same sequences of cells that are active on the track are active, but they're compressed in time. And so essentially, while a group of place cells or ensemble of place cells may be active in this sequence that unfolds over seconds, during sharp wave ripples, the same sequence is replayed over, let's say, tens of milliseconds to hundreds of milliseconds. And so what's really interesting is how does the hippocampus generate these sequences in multiple time scales? And what's particularly intriguing is how does it do this without connections between pyramidal cells? Because unlike most cortical areas, Ca1 pyramidal cells do not synapse onto one another, which makes it very hard to imagine that it's something like a synfire chain activating these sequences. So if we believe that the hippocampus, and in particular Ca1, is generating these sequences, what is the circuit that actually has to do this job? And so this is like a canonical microcircuit in Ca1 that demonstrates that although pyramidal cells don't synapse on each other, they do actually interact with each other disynaptically by capturing, which, well, you'll see why I use that word later in the talk, but capturing the attention or driving spiking activity in interneurons, which synapse on their peers. And so in this cartoon, we're thinking about two assemblies of pyramidal cells, the green and the gray, 1 and 2. And we're thinking about how they could be interacting. So it could be the case in this cartoon that assembly 1 excites an interneuron that then inhibits itself and also other assemblies. So this can be thought of as like a lateral inhibition motif. There's a lot of evidence that this is actually the case. This competition for representations in Ca1 is true. And one of the most, one of my favorite examples is from David Dupre's lab at Oxford, where they used optogenetics to selectively manipulate the activity of one assembly and then to observe how the other assembly reacts. So in this experiment, they expressed an inhibitory opsin, which is ARCH-T, in pyramidal cells of a certain assembly, so encoding a specific place. And in these raster plots here, what you see is the light is this yellow area, and you see that the cells that express the green light, unsurprisingly, stop firing when you turn on the light. And also unsurprisingly, they also lose their place fields. So you see the two columns on the left where it says 6.3, 3.8, 4.6, those are the peak firing rates in the place field. On the right, the right column of 3 circles, that's when the light is on, and therefore obviously the place fields are gone. So this isn't it's surprising. You turn off cells, they stop spiking, right? But this enables you to really test the hypothesis that these cells were initially active because they were inhibiting competing assemblies via these interneurons. So what happens to assembly 2 when assembly 1 is inhibited is a really important test of what's going on. And amazingly, and in support of this circuit motif being highly active or highly important for Ca1 activity, is that the pyramidal assembly 2 drastically increases its firing rate essentially immediately after you turn off the first assembly. So it's not that you needed to learn something new or this takes a very long time. Essentially, instantaneously, when one assembly is turned off, another one pops into activity in its place, suggesting that it was actually inhibited by that first assembly. But of course, this activity doesn't have to be anything useful to the animal, right? So you're turning off a whole bunch of neurons, a bunch of other neurons are becoming active. This could obviously result in pathological activity or seizures, or who knows what this activity is going to actually represent. It turns out that none of that actually happens. They actually form a new map that looks, in terms of the tuning of the place fields, very much like the original map. So this seems like a physiologically relevant manipulation, but it's a big puzzle. So how exactly does this happen? So we know there's near zero pyramidal connections, and this cartoon is what we assume is happening. So the assembly interaction through engagement of lateral inhibition. So this seems like pretty conclusive evidence that this is actually going on. And so how does this actually work at the synaptic level? And that's what we were really interested in, and that's what we use the micro LEDs to address. And so how can we study this synapse here in the red circle? So what we're really interested in studying is, because we know this competition between assemblies is so important, we want to understand how assemblies, such as the green one here, how do they capture these interneurons? How do they, in terms of making them fire when the pyramidal cell spikes, how does that engage an interneuron, make it spike to inhibit a competing assembly? So we really want to understand the synaptic organization of the inputs from pyramidal cells to interneurons in Ca1. And we also want to understand the short-term and long-term plasticity. And during this talk, we're going to talk mostly about the organization and the dynamics of these synapses, not considering how this relates to place tuning and learning. In the next part of this series, Sam McKenzie is going to talk about how the place tuning of cells in Ca1 and the plasticity relates to these interneuron and pyramidal cell interactions. But for now, this is looking like a deep dive into the organization and dynamics of pyramidal cell interneuron organization. And we're going to use these micro LED probes to take an unprecedented look at that circuit. Typically, though, and people such as myself have used acute slice preparations to look at synapses between two cells, right? The standard sort of for looking at the physiology of a synapse is to make a paired recording between the presynaptic and the postsynaptic cell, as you can see here. And you could inject current through your recording electrode into the presynaptic cell to make it spike, and then you can observe the EPSC or EPSP in the postsynaptic cell and confirm that these two cells have a synapse between them and study its dynamics. This isn't really feasible in vivo for many, many pairs of cells. So there's another feature of these experiments that we want to take advantage of, illustrated here. And so in the top here, you see the presynaptic cell is again being made to spike by injection of current. But here, this time, the postsynaptic cell is depolarized by current injection. And so they're depolarizing it such that it fires at some not too high, not too low rate, sort of a medium rate that's variable throughout the spike train. And what they're going to try to see, which you can see in the bottom where it says spike transmission very clearly, is what is the effect of the presynaptic cell spiking, not on the membrane potential of the postsynaptic cell, but the probability that it fires within a certain temporal window around the presynaptic spike. And so what you see here is that very reliably, from this really nice paper from Galarreta and Hestred, is that the spike in the presynaptic cell reliably increases the spike rate of the postsynaptic cell. And so since we can't do this experiment for more than 2 cells, especially in vivo, we wanted to test whether this is really, you could actually use just the spike transmission, so the probability of one cell spiking after another, if we can use that as a way to detect synapses and to study their dynamics. We weren't the ones who invented this technique. People have been trying to look at the cross correlations, as we call them, the temporal relationships between the spike trains of 2 neurons. So since the 60s, people have been looking at these, using these kinds of analysis to try to infer information about the function of the circuit. But I think there's, it's safe to say that some people, I think, tacitly assume this is fine and believe that you can use this to find a connection, and other people are probably not so sure. So the first question that we want to use the micro-LEDs to address is, can synapses be inferred from spike transmission? And why is this so important? So what would be so crucial if we could do this? Well, what we could do then is we can record hundreds of neurons with a silicon probe. So if we could detect just using the spike times connections between cells, that would enable us to look at a very high throughput, in a very high throughput manner, look at the connectivity. And so this is a picture from one of Juxtacellular's papers from the silicon probe. This is showing where we're going to be placing these probes across the pyramidal layer. And in the bottom, you see this is the totally raw voltage from one of these micro-LED probes. And this is 6 traces of that activity. And in the bottom, you can see the rosters of these cells. So we can easily pull out, this is a standard technique. We could pull out the spike times of many neurons. Here there's only 4, but in reality, you can record up to hundreds at a time. And then we wanted to use this rich data to see, to study these connections. But first, we have to see how can we actually determine their connectivity. In this plot, what I'm showing is this cross-correlation relationship, so the timing relationship between a pyramidal cell and an interneuron. So this is trying to look at that synapse, again, that I keep saying is so important. On the left in black, this is 4,000 spontaneous pyramidal cell spikes overlaid on top of one another. And then at the same time, the spiking of an interneuron during the spontaneous time also, and when those spikes occurred in relation to that pyramidal cell spike. And what you can see is that about a millisecond after the pyramidal cell spikes, there's this significant density in the, I just got a text saying the titles are being cut off in my slides, but I don't think, I don't think that's on my end. I don't think I can control that. So I'm going to continue with the talk. So the title of this slide is, how can we detect, or can we detect excitatory synapses using spike timing? So in any case, I said on the left, this is an overlay of thousands of pyramidal cell spikes, and then the interneuron spikes seem to cluster right after that. So does that mean that the pyramidal cell is releasing glutamate and making that interneuron spike at this synaptic conduction delay? You could quantify this effect looking at the relative timing of the 2 cells. On the right, this is the same 2 pair of cells, and you can see that after the pyramidal cell spikes, which is time 0, the interneuron is much more likely to spike about a millisecond later, just that histogram. So what we wanted to know is, can you calculate the spike transition probability from this increase, this excess synchrony at this delay between the 2 cells? And that, if that's true, that would mean that if every time you see this kind of temporal relationship, does that actually mean that there's a synapse there? The alternative would be that this synchrony is produced by some other features of the network. So common inputs to both cells, there's all sorts of ways you could imagine in different circuits getting this kind of synchrony. But we wanted to test to see if this, which one of these is the case. So this slide is titled Disengaging Presynaptic Spiking of Single Pyramidal Neurons from Network Timing. And this is essentially the key to how we're going to use these the micro-LED probes is we're going to try to stimulate just the one pyramidal cell, or as few pyramidal cells as we can, to obviate the effect of the network, because we're going to stimulate at random times. So it's not that we're going to let the network dictate when the spikes occur, because that could have obviously physiological synchrony, but we're going to control when the presynaptic cell spikes, and then we're going to see if you still see that temporal relationship. And so the hypothesis is that we still see that temporal relationship, then that means that this is a synapse, because we're controlling the spiking. Essentially, we're trying to reduce the preparation as much as we can to just these two cells. But of course, this is all in vivo in a freely behaving mouse, either in a home cage, awake and sleep data all combined, I should say. So again, this is for my paper with Sam. So what we did is we put micro-LED silicon probes, as I said, in the hippocampus of freely behaving mice. And what you can see on the right is this is a single pyramidal cell, and this is its response to light. So it's very easy to robustly activate pyramidal cells. I should say this isn't a mouse. It's a CAM kinase Cre mouse, so it expresses the Cre recombinase selectively in excitatory neurons. And Ca1, that's the pyramidal cells. And this is crossed with a mouse, it's the Allen Institute AI32 mouse, which has channel rhodopsin, except it is in a certain orientation with LOX-P sites, such that it doesn't actually express channel rhodopsin unless there's Cre also present. And so we crossbred these mice, resulting in channel rhodopsin only in pyramidal cells. So, which has been verified before that this works. So like I said, you can, on the right, you see this plot. These cells fire more with light, but how specific is it actually? So how do we know how many cells we're firing? We're firing off the light. So on the bottom left here, you see direct excitation of single unit pyramidal cells. So what we're doing here is we're looking at one pyramidal cell of the many we're recording, and we're seeing how it responds to stimulation of different shanks of the silicon probe. And so you can see what the effect essentially is. This cell is recorded on shank 3, and essentially light, we did 10 different stimulation intensities, and you can see that the shining the light on shank 1 or shank 2, and basically shank 4 has very little to no effect. In shank 3, there is a strong effect of the light, making this cell fire more, so the firing rate is getting color, and that the latency to spike decreases with increasing intensity, as you would expect for directly depolarizing a neuron such as a pyramidal neuron. The middle plot is showing how the relationship between intensity and the firing rate gain of these cells. So these are single unit spikes. And importantly, so what this demonstrates that the micro-LEDs can do is that you could parametrically control the timing and the rate of small groups of cells. This is very good towards our goal of trying to disengage small groups of cells from the network activity. And on the right, what you can see is that it turns out that this is through all of our experiments. This is how many neurons were active elite to over 50% of their baseline firing rate across all experiments. And so what you can see is some of the experiments we were stimulating maybe 20 cells at a time, but that was a minority. Most of the time it's less than 20 cells, and a lot of the time it's far less than that. It's in Ca1, because of the dense packing of the cells and the fact that the opsins are expressed all along the dendrites of the pyramidal cells, not just in the soma, makes it currently extremely difficult in Ca1, at least I should say, to stimulate single neurons. And so that's something I could talk about more in the question and answer if people are interested exactly the details of that and some of the solutions that people are trying to come up with. But we were very happy that these micro-LED probes allowed us to have this level of control, which isn't actually possible with other techniques to simulate this small number of neurons in freely behaving animals. So what we wanted to do is compare the spike transmission for spontaneous and evoked spikes, obviously. So this would be the spontaneous case. This is the evoked case. So we're evoking spikes at random times. If you look at the middle plot here for a second first, These are those CCGs again, but this is with the baseline subtracted. And what you can see is in gray is a spontaneous cross correlation. So you see that there's excess synchrony at a short delay, about a millisecond after the pyramidal cell spikes. This is the interneuron spiking. And then the evoked pyramidal, this is the evoked pyramidal cell spikes. They also produce spikes in the interneuron at about the same delay. So this looks very, this is for about 120, 30 cells. These are the CCGs, but now each row is a CCG, and the y-axis is now in color. For, you know, if it was the middle plot, the y-axis is now in color, and this is for spontaneous and evoked. And what you see is there's essentially no pairs for which, you know, there's no pairs for which there's a peak on the left and not on the right. There's essentially a very nice correlation between the evoked transition probability and the spontaneous transition probability, which is shown in the plot on the right. It's slightly shifted towards the evoked actually being higher because the pyramidal cells are actually more synchronous than normal because of our stimulation, which boosts the spike transmission from each cell, which is something that we actually studied and quantified, and we'll talk about it in a minute. We wanted to verify this with a second technique, so we actually used a technique called Juxtacellular Recording, where you stimulate a single cell with a glass electrode, and then we also, with a second silicon probe, a different silicon probe, recorded the activity of the local neurons, of the interneurons, and you can see here it looks very similar to the data we collected with the micro-LED probes. I should say that another question that I imagine people might have is how do we know from these extracellular recordings which cells are interneurons and pyramidal cells? And that's another thing I can address later on. But essentially, we were very happy that these micro-LED probes allowed us to say that when we see this kind of temporal relationship, it actually is indicative of a monosynaptic connection and not network coordination or anything else you might imagine. And so with this confirmation in our hand, we were able to look at a lot of features of the Ca1 network. So for the rest of the talk, I'm going to go over these properties of the pyramidal cell interneuron circuits in Ca1 and talk about their organization and their short-term dynamics. And like I said, this is a very good setup for the talk that you're going to hear in the next session, the next iteration of this from Sam McKenzie. And so what this cartoon is showing that the probes of the shanks of the silicon probe are in gray here. The white squares are the recording sites. The colored triangles are the pyramidal cell putative somatic location. It's just which recording site had the biggest spike. That's how they're placed vertically. The horizontal placing is, we know which shank they're recorded on, but the jitter, sorry, the x-axis jitter within shank is random just for visualization. The lines between the pyramidal cells and interneurons are in color. The color signifies the transmission probability. And a couple things become really apparent when looking at this, one of which is that there seem to be interneurons, some of which they get very little inputs, and there seem to be the other interneurons that get tremendously numerous and strong inputs from many of their peers. And so we wanted to, using this connectivity, we wanted to quantify these things. So what is the synaptic organization like? So one thing we wanted to know is how many interneurons does a pyramidal cell actually make contact with? And it turns out that this is it's a relatively normal looking kind of distribution where most pyramidal cells have few postsynaptic partners. And but then when you look at it the other way, it turns out it's something very interesting where if you look at the number of, if you look at how many presynaptic partners different interneurons have, it looks like it's this very skewed distribution where many interneurons only have a few presynaptic partners, but some, like in this previous slide we saw, like the one on the top right, this orange cell that gets input from all these other cells, some interneurons get inputs from essentially every pyramidal cell we recorded. And so this is something that we're really interested in, is how these differences are going to be related to things like tuning. So this was a sort of an unexpected result. And another thing we looked at is, in Ca1, there isn't a local clustering, like a spatial anatomical clustering of cells with common tuning. So what I mean is that if you record two place cells that are right next to two pyramidal cells that are right next to each other, their place fields are not necessarily more similar than pyramidal cells that are physically farther apart in the brain. But of course, axons are only so big or so long, and we wanted to see what the connectivity was like with the pyramidal cell interneurons in these, are there like functional local areas in Ca1? And what we observed is that it turns out that the local connections are more numerous, and they're also a lot stronger. And so there seems to be a spatial limitation in terms of how far a pyramidal cell can reach to control an interneuron. So that was sort of the structural side, that we're starting to see these different patterns or statistics of these connections. But what about the dynamics, right? So we want to understand how a pyramidal cell can activate an interneuron in service of inhibiting a competing cell. But what if the pyramidal cell is just fired? Is it going to be able to inhibit that, suppress that competing assembly every time it spikes, or does the spiking history of the pre or even the postsynaptic cell affect this ability? So typically, this is a plot from a review paper that, typically you would study synaptic facilitation or depression using an intracellular or whole cell recording, and you do a train of stimulations, as you see here, and you would see whether the responses went up. So on the top, you see that's facilitation. Do they go down? Is that depression? Or do they not care? Or are they static? So we wanted to ask, can we see the same kind of dynamics for naturally occurring spike trains in the awake hippocampus? And so we did this by assaying the probability of a pyramidal cell evoking a spike in an interneuron conditioned on the time since the pyramidal cell last spiked. So here, this ISI, we're looking at how the probability of AP2 in this little cartoon, of how the probability of AP2 to make the probability of a spike in the interneuron based on the time since AP1. And so very interestingly, we were very excited to see that you do see the short-term plasticity dynamics. So this is the change in transmission probability, which, if we go back for a second, just to clarify this for everybody, essentially that's the change. It would be the change in the height of this middle plot. So the spontaneous and evoked here in the middle. If this peak gets higher, that's an increase in transmission probability, which we're speaking of as a proxy for synaptic strength. And if this transmission probability goes down, that's a weakening of the connection. And so, as you can see here, the top, the x-axis is the presynaptic ISI. So on the left is shorter, it's a high firing rate, shorter. The right is longer. And so the top is an example of a depressing connection, where as you fire faster and faster, you get weaker. The bottom is the facilitating connection, where if you've just fired this next spike, so AP2 here is going to have a higher probability of making that interneuron spike. What about the other way? Like we said, we're looking at what if you're an interneuron and you're having different pyramidal cells connect to you? Are all of those connections going to be the same direction? So in the top here, we see that one pyramidal cell can input to two different interneurons, and the dynamics of those synapses can be different. One could be depressing, and one could be facilitating. What about for interneurons? Are all the interneurons having the one type of inputs? And it turns out that's not the case, that individual interneurons can have different kinds of relationships. And so this is one interneuron here that has three different presynaptic partners. And what we're seeing is that each synapse has a different dynamic. So the dynamic is synapse specific, not cell specific. So it's cell pair specific effectively. And so this is just to illustrate how drastically different this can be for different inputs of the same cell. So this is a single interneuron, and this is the plasticity, short-term plasticity curves for 21 of its inputs. and you can see they're quite variable. And this is for a larger number of cells. It's split versus the depressing, the facilitating. Essentially, what you can see from the depressing, FS is fast spiking. We looked at compared to the interneurons just by fast spiking and non-fast spiking. And essentially, you see that you see facilitating and depressing connections in both fast spiking and non-fast spiking cells. You can clearly see that most of the connections that we've detected are depressing, but that it's quite variable. So this was really interesting and something that has really stuck with me, that the plasticity is specific to the connection, meaning that if you're a pyramidal cell, your spike train is going to be read out differently by different downstream readers, depending on the dynamics of the synapse and the statistics of your spike train. But what about the postsynaptic cell? How does the firing rate or the timing of the interneuron actually determine its receptivity to being made to spike by the pyramidal cell? And so to do that, what we're going to do is we're going to assay how many spikes, first to look at the rate, how many spikes did the interneuron fire in the time before the pyramidal cell spike that we're going to examine the spike transition probability of. And it turns out that the rate actually really doesn't matter. And so in the top here, this is the raw data for epochs where the interneuron was firing at a certain rate before the pyramidal cell spike. And obviously, we're pre-selecting these rates, so the overall peak in the CCG is higher. But when you subtract out this background to look at the excess spike transmission, or the excess synchrony, meaning the spike transition from this one pyramidal cell, you see that the peaks are all about the same height. And on the right, you can see that after you remove this baseline, which doesn't have to do with a single pyramidal cell, that the rate of the postsynaptic cell actually doesn't matter. But it turns out what really matters is the timing. And so now we're going to assay if you're a pyramidal cell spike, does it matter? Does the probability of you making the interneuron spike, does that probability change depending on when the interneuron last spiked? So before we looked at when the pyramidal cell last spiked, so its spiking history, but how does the spiking history of the interneuron affect its ability to be made to spike by the pyramidal cell? And it turns out the timing is super important. And here, this is in green, what you see is this spontaneous data, so all the spontaneous recordings. And you can see that there's an optimal ISI. So a pyramidal cell is most able to drive an interneuron to spike if the interneuron just spiked about 20 milliseconds ago, so about a gamma cycle previous. But we really wanted to know, or we really wanted to know whether this was a property of these two cells, so whether this effect would exist in isolation or if it was like a network dynamic. So to do this, we used the we used the silicon probes to control the activity and to make sure that we weren't just tied, to randomize it again, till we weren't tied to the network. And what you can see for the evoked spikes in blue, this curve looks very similar, and the peak is in the same place. So this could be due to many things, including intrinsic resonance of the interneuron, so the postsynaptic cell membrane, which is been observed to be resonant at about this gamma frequency. So the postsynaptic spiking is really important for influencing the receptivity and the routing of information from pyramidal cells to interneurons. And remember the beginning when I said that, when we did the optogenetics simulation, the spike transition was a little bit stronger. And that was because we weren't able to stimulate single cells the whole time. So we wanted to really look at this effect. So if you're a pyramidal cell and you spike, what is the probability of making an interneuron spike if another pyramidal cell that's also presynaptic to the interneuron spikes at different delays to you, or if another pyramidal cell that does not input to that interneuron, what is the effect of that spiking at different delays to you? And the difference, that would, obviously if there's 3 cells for the independent input existed in isolation in the universe, there wouldn't be any effect, but obviously this is part of a larger network of interacting neurons, so that could produce an effect. So first I wanted to say do pyramidal cells that input to the same interneuron, do they themselves tend to fire together more at different times, at different ISIs? And it turns out that this is true. So in green on the top is the count of, excuse me, so it's the synchrony of, sorry. The pyramidal cells that share common inputs, share a common postsynaptic target in green are more synchronous with each other, especially shorter time scales, than pairs that do not synapse on the same interneuron shown in black. So there is this functional implication of this, as we're like, so you can imagine, the cells that input to the same interneuron might be part of the same assembly, like we talked about originally. So here we looked at exactly how this ISI between the two pyramidal cells affects the spike transmission. And so we looked at this for both the convergent and independent inputs. And you can see that the independent line isn't flat, but that's because of the network effects. But what you see in the cells that are convergent is that you have this really strong gain in spike transmission probability, especially at short time scales. So it is the case that if two pyramidal cells fire close in time to each other, that they are able, or they appear to be able to more strongly drive this interneuron than if they fire in isolation, temporally isolated. So again, this is all spontaneous data recorded from a freely behaving mouse, and sometimes they're sleeping, sometimes they're doing stuff. We wanted to know, is this an artifact? So is this actually physiology of the circuit that we're observing? Or does it have to do with a network mechanism or a behavior or sensory input of something else that's creating this effect? And so again, we turn to the optogenetics, which is why these probes are so great. We could do these manipulations where we control a small number of cells. And what we actually see, this is again, so gray is spontaneous. This is now both for the convergent. And then when we stimulate, you see that, the artificial spike train is the Evoked spikes in blue. They also have an increase. So it isn't just, so it seems that it's not just the network effect, that you do have this boost in gain even for spikes that are controlled by us. So it's not that there's not anything unobserved going on. It seems that a large part of this effect, at least, is due to just considering these three neurons. You can explain this. So this seems to provide some kinds of temporal rules for these assemblies to capture the activity of these interneurons. I've been saying the whole time, in service, we think of have these competing assemblies, as that behavioral data showed before. So I'm quickly just going to go over the conclusions. This is all from my paper with Sam, it was 2017. And so the first thing we did, the first conclusion is that we can detect in Ca1 of the hippocampus, we can detect monosynaptic connections using spike timing, and that presynaptic cooperativity boosts synaptic drive, specifically at very short timescales. These connectivity motifs are inhomogeneous, like we saw the skewed distributions. And importantly, short-term plasticity is connection-specific, not cell-type specific, and you see both directions. And that the synaptic drive is more modulated by the postsynaptic timing than by rate. So how fast the postsynaptic interneuron is firing at the time the pyramidal cell spikes doesn't matter that much. It just matters when it last spiked. Sorry about that. And so this is not this is unpublished data. So this is all these micro LEDs that we're really excited about that Sam and I did, is that we tried to look at whether we could examine the plasticity of these connections. So excuse this slide for being a little bit busy, but so essentially we're looking at 4 cells that we recorded with the silicon probe, and we're looking at the temporal relationships between these cells, so the connections, and then how they change after we pair the activity of one pyramidal cell with spiking in the interneuron. So a standard forward timing, spike timing dependent plasticity protocol would be pre-fires, then post-fires. So but we're not able to control both types of cells in this experiment, but what we can do is we can express channel rhodopsin in interneurons. These are PV parvalvein expressing interneurons, and they'll express channel rhodopsin in a transgenic mouse. And on the upper right, what we can do is we can wait until a pyramidal cell spikes. We can do online spike sorting, detect that spike, and then as soon as that cell spikes, we can then, within a few milliseconds, stimulate the PV cells. And when you see in the histogram below that little cartoon on the top right is we can see that the interneurons do robustly spike a lot more right after this pyramidal cell. So we can generate forward timing STDP protocols using closed loop stimulation, online spike sorting, and combined with the micro LEDs. If you look at the middle row on the left, so the correlation of light onset with the spiking of the presynaptic cells, it turned out in this session, our detection of the cells wasn't actually perfect. So we were actually stimulating after two of the three cells were spiked. So pyramidal cell 1 did not trigger light. Pyramidal cells 2 and 3 did trigger the light both together. And what we can do is we can compare the spike transition probability before and after we did this forward timing protocol where we stimulated the interneurons after spontaneous spikes of pyramidal neurons 2 and 3. And very interesting, what you see is the before pairing is black, the after pairing is in red, is the pyramidal cell 1, that connection actually appeared to get weaker. So the spike transmission probability went down. But pyramidal cells 2 and 3, these are the paired ones, increased, which is very interesting and suggestive that these synapses do undergo forward timing at least STDP. And this is quantified for a relatively small number of cells, but it's significant in this plot below that the red little diamonds, excuse me, or what just the postsynaptic cell, and then the pre-post pairing was the black. And so you can see that the slopes are different in this plot. The spike transition probability is higher for these cells that we paired overall. But obviously this is preliminary. But it's another, I think, another nice example of the power of these experiments. You can, stimulate a small enough brain area that you're not creating non-physiological conditions. You can really look at the functional interaction between small groups of neurons. And so with that, I'd like to acknowledge the members of my lab at Virginia Tech, Kaiser, Earl, Leon, and Reagan, and also, very importantly, Yuri Bujaki at NYU, where this work was done, and Sam McKenzie, his lab is at the University of New Mexico, and also, of course, Yusik and Khan from University of Michigan. And thank you, and I'm happy to take any and all questions.